Hello बच्चों, welcome to SPSVERSITY. In the previous session, we discussed about mitosis, that is the cell cycle. Cell cycle, karyokinesis, cytokinesis we discussed. Now we are going to discuss about meiosis and sexual reproduction. So meiosis में क्या होता है? So in meiosis, the same way we'll have interphase like cell cycle. In meiosis also we'll have a interphase. Along with interphase, there is meiosis 1 and meiosis 2. Okay. So along with interphase, there is meiosis 1 and meiosis 2. After this, there will be a cytokinesis. A meiosis 1, it is very important phase in the entire meiosis. So in the entire meiosis, it is a very important phase. So in meiosis 1, the chromosome number becomes half. Okay. So here if you look into this meiosis, there are two divisions. इसमें two divisions रहता. Meiosis 1 और meiosis 2. Meiosis 1 में chromosome number it will become half. It will become half. If you look into meiosis 1, इसमें 4 phases रहता. So prophase 1, metaphase 1, anaphase 1, telophase 1. और meiosis 1 के बाद में cytokinesis 1. So the entire meiosis 1 can be divided into 4 phases: prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis 1. So this the longest phase is prophase 1. Longest phase prophase 1. So depending upon the changes, prophase 1 में 5 stages रहते. So depending upon the stages, इसमें 5 stages है. So क्या है ये लेप्टोटीन, जाइगोटीन, पैकेटीन, डिप्लोटीन, डायकिनेसिस. So लेप्टोटीन में क्या होता है? So it is the first phase of prophase 1. इसमें condensation of chromatin material. When chromatin material is condensed, it will form chromosomes. So chromatin material से क्या form होता है कि chromosomes. Okay. So एक chromosomes में homologous chromosomes रहता. So homologous means chromosome from one parent, that is from male it is called paternal chromosome, और फीमेल से आने वाले क्रोमोसोम को क्या बोलते हैं कि मैटरनल क्रोमोसोम. एक पेयर रहता, एक पैटर्नल क्रोमोसोम, एक मैटरनल क्रोमोसोम. So these homologous chromosomes start pairing. So इसको क्या बोलते हैं कि synapses. पेयरिंग के लिए एक प्रोटीन चाहिए, प्रोटीन कॉम्प्लेक्स, इसको क्या बोलते हैं कि synaptonymal complex. The synaptonymal complex will hold the homologous chromosomes close to each other. ऐसा दो chromosomes, one paternal chromosome और one maternal chromosome, ये दोनों कंबाइन होता है, एक पेयर. सो एक पेयर को क्या बोलते हैं कि बाइवैलेंट. बाई मीन्स दो, इसमें दो क्रोमोसोम्स रहते हैं पेयर में, इसमें दो क्रोमोसोम्स रहते हैं. तो जाइगोट में क्या होता है कि सिनाप्सिस. सिनाप्सिस के वास्ते टू क्रोमोसोम्स कंबाइन बाइवैलेंट फॉर्म. एक बाइवैलेंट में दो क्रोमोसोम्स रहते हैं. जाइगोटिन की बाद में नेक्स्ट स्टेज है कि पैकेटिंग, ए बहुत इंपॉर्टेंट फेज. ये पैकेटिंग में नॉन सिस्टर क्रोमैटिड्स क्रॉसिंग हो रहा था. नॉन सिस्टर बोले तो क्या है? क्रोमैटिड्स ऑफ बोथ क्रोमोसोम्स, इसको क्या बोलते हैं कि सिस्टर. क्रोमैटिड्स ऑफ डिफरेंट, दैट इज वन मैटरनल और पैटर्नल, इसको क्या बोलते हैं कि नॉन सिस्टर. तो इसमें सिस्टर और नॉन सिस्टर क्रोमैटिड्स रहता हो. सिस्टर बोले तो क्रोमैटिड्स ऑफ सेम क्रोमोसोम. नॉन सिस्टर बोले तो क्रोमैटिड्स ऑफ डिफरेंट क्रोमोसोम. दैट मींस वन मैटरनल क्रोमैटिड, वन पैटर्नल, इसको नॉन सिस्टर क्रोमैटिड्स बोलते हैं. ये नॉन सिस्टर क्रोमैटिड्स के बीच में क्या होता है कि एक प्रोसेस होता है, जिसको क्या बोलते हैं कि क्रॉसिंग ओवर. इसको क्या बोलते हैं कि क्रॉसिंग ओवर. क्रॉसिंग ओवर में क्या होता? सो क्रॉसिंग ओवर में exchange of chromatin material takes place. So crossing over में क्या होता है कि exchange of chromatids. So this point where crossing over takes place, इसको क्या बोलते हैं कि chiasma. Exchange of material, after exchange of chromatin material, it leads to variations. After many variations, it leads to formation of new species, that is speciation. So when you come to evolutionary point, crossing over plays an important role. So the basic natural process for variations is crossing over. So which takes place in पैकेटिंग. ये प्रोसेस पैकेटिंग होता है. एवोल्यूशन के लिए बहुत इंपॉर्टेंट नेचुरल प्रोसेस. So you can see in the picture. So this is chromatids. So these two non-sister chromatids. These two are non-sister chromatids. A non-sister chromatids के बीच में crossing over हो रहा है. This is point where crossing over. So ये point को क्या बोलते हैं कि chiasma. ये point को क्या बोलता? chiasma. आप पिक्चर में देख सकते हैं आफ्टर क्रॉसिंग ओवर ये दो क्रोमैटिड्स में this part of chromosome has been exchanged with this part of chromosome. Just it is an exchange of chromatids. So which leads to variations. So which leads to variations. Okay. So what is actually meant by crossing over? So it is just of exchange of a part of chromatids of homologous chromosomes. So you can see in this picture, this part of chromatid belongs to this chromosome. This part of chromatid belongs to this chromosome. Both are exchanged. So a process को क्या बोलते हैं कि crossing over. So crossing over variations के लिए बहुत a basic natural process. Due to this exchange of this, there is a change in the pattern of genes. So you can see in this picture. So whatever genes are present on this DEFG, it has come to this chromatid. Okay. So due to this crossing over, it leads to genetic recombination. Genetic recombination means formation of new combination of genes other than the existing one. When new combination of genes are produced, so what happens is there is no longer contain identical genetic material. उसमें identical genetic material नहीं रहता, which leads to variations. So this new combination of genes को क्या बोलते हैं कि variations. Finally, which leads to speciation, that is formation of new species. So it will show an impact on change in the organisms. Like a lot of variations, many changes from one generation to other, which leads to adaptations like this. Many changes are observed due to these variations. So packaging is a very important phase where crossing over takes place. Okay. The next stage क्या है कि diploting. So once crossing over is completed, so what happens is the chromosomes start repelling with each other. That is, homologous chromosomes start repelling with each other. They start separating. And this separation reaches to maximum in diakinesis. That is, in the last stage of prophase 1, the synaptonymal complex will disappear. और इसके साथ nuclear membrane और nucleolus भी disappear होता है. So in diakinesis, the homologous chromosomes start separating and synaptonymal complex disappears. इसके साथ न्यूक्लियर मेम्ब्रेन और न्यूक्लियोलस भी डिसअपीयर होता है. So this is about prophase 1. So अभी तक हमें पढ़ा है prophase 1. इसमें 5 स्टेजेस रहता. लेप्टोटीन, जाइगोटीन, पैकेटीन, diplotin, diakinesis. लेप्टोटीन में क्या होता है? कंडेंसेशन ऑफ क्रोमैटिन मटेरियल. जाइगोटीन में सिनेप्सिस. पैकेटीन में क्रॉसिंग ओवर. डिप्लोटीन, chromosomes repel with each other. Diakinesis, chromosomes separate from each other. Next, the nuclear membrane, nucleolus and synaptonymal complex will disappear. A prophase 1 is the longest phase. After this, it enters into metaphase 1. A metaphase 1 means so pairs of homologous chromosomes move to center of the cell. Center of the cell means इसको क्या बोलते हैं कि equator. They will reach to equator. So all homologous pairs of chromosomes are arranged in the center of the cell. इसको क्या बोलते हैं कि equator. वो equator में align होता है with the help of spindle fibers. Okay. Meta means middle of the cell. Middle of the cell में chromosomes align होता है. The next phase is anaphase 1. So in this anaphase 1, the homologous chromosomes are completely separated. So इधर homologous chromosomes पूरा separate हो जाते, अलग हो जाते. Due to this process, what happens is the diploid number changes to haploid. Diploid क्या होता है कि haploid. Okay. So here diploid number of chromosomes becomes haploid. So here you can see in the picture. So this is one homologous chromosome with two chromatids. Okay. So these homologous chromosomes are separated completely, and this process is called disjunction. इस process को क्या बोलते हैं कि disjunction. Due to this disjunction, what happens is diploid has become into haploid. So chromosome number becomes half. So this is anaphase 1 is the phase where chromosome number becomes half. Diploid will become into haploid in anaphase 1, and that process is called disjunction. So that process is called disjunction. anaphase 1 के बाद में telophase 1. A telophase 1, which is quite opposite to prophase, where the nuclear membrane is formed, nucleolus is formed. At each pole, the surrounding two chromosomes, nuclear membrane and nucleolus is formed. After telophase 1, the division of cytoplasm takes place, that is cytokinesis, which divides into two cells. So that is after anaphase 1, from one cell, two cells are formed. And these are having only half of the chromosome. You are having only half of the chromosome. So इससे meiosis 1 एक complete हो गया. Meiosis 1 is completed. So in meiosis 1 we studied prophase 1, metaphase 1, anaphase 1, telophase 1. इसके बीच में meiosis 1 और meiosis 2 के बीच में chromosomes replicate नहीं होते. There is no replication of chromosomes. Okay. So the next is meiosis 2. After completion of prophase, that is the meiosis 1, the next phase is meiosis 2. A meiosis 2 is equivalent to mitosis. It is just equivalent to mitosis. From two cells, four cells are formed. Same like mitosis. Where इसमें चार stages prophase 2, metaphase 2, anaphase 2, telophase 2. The same changes. A new spindle forms around chromosomes. Nuclear membrane and nucleolus will disappear. The same things. Chromatin material will condense and form chromosomes. बाद में क्या होता? Metaphase 2. Chromosomes lying along the equator, they're attached to, that is, spindle fibers are attached to centromere. A metaphase 2 में होता है. बाद में क्या है कि anaphase 2, where centromere will divide. Due to this, chromatids are separated and which move to opposite poles. Anaphase 1 और anaphase 2 में रहते. Anaphase 1 में division of centromere नहीं रहता. सिर्फ anaphase 2 में division of centromere होता. Due to this, chromatids are separated and these move to opposite poles. The same thing in telophase 2. Nuclear envelope and nucleolus is formed and chromosomes undergo decondensation. So finally, हमें कितने cells आते हैं कि 4 cells from one cell will get 4 cells. Is 4 haploid cells. इसमें एक सेल से 4 सेल्स आता है और उसमें क्रोमोसोम्स नंबर सिर्फ आधा रहता, पेरेंट सेल से आधा रहता. Half of the chromosomes from the parent cell. Okay, so here you can see this is one parent cell. This is after meiosis 1, 2 cells are formed. After meiosis 2, इसमें कहाँ से होते थे? From each cell will get 2, total will get 4. Okay. Meiosis में during crossing over we can see genetic variation. Okay. This genetic variation is having a lot of importance. There are 3 mechanisms to form genetic variation. One is independent assortment. Independent assortment means separation of alleles independently during formation of gametes. Crossing over और बाद में random fertilization. Fertilization is not a predetermined process. This is random. It may take place between any male and female gamete. It's not a predetermined one. Due to independent assortment, crossing over or random fertilization के वास्ते genetic variations are formed. So independent assortment means random distribution of homologous chromosomes during meiosis. Even fertilization of egg, that is female gamete and sperm, is random. Okay. That is, if we take the square of 223 is 64 trillion. Okay. So the genetic combinations can occur in gamete, that is, it is not limited, it is practically unlimited. And when you come to this genetic variation importance, there is a lot of importance in the entire life of living organisms. If you take our ancestors and the present generation, there are a lot of their changes. Okay. The changes, the reason for changes is meiosis and random fertilization. And these variations will increase, these changes will increase from one level to other, that is from one generation to other generation. So natural selection does not always favor this one. And when it comes to meiosis, meiosis is having an importance, that is in the sexual reproduction. In sexual reproduction, gametes are formed, male and female gametes form both. So when you take in humans, formation of male gametes को क्या बोलते हैं कि spermatogenesis. That is, male gametes are formed from testes. उसको process को क्या बोलते हैं spermatogenesis. स्पर्मेटोजेनेसिस में पहला सेल डिप्लॉयड सेल उसको क्या बोलते हैं कि स्पर्मेटोगोनियम. सेल का नाम क्या है कि स्पर्मेटोगोनियम, which is diploid. This spermatogonium will enlarge and form primary spermatocyte. This primary spermatocyte will undergo meiosis 1, which will form two haploid cells. The primary spermatocyte से दो haploid cells form. उसको क्या बोलते हैं कि secondary spermatocytes. The secondary spermatocytes again undergo meiosis 2 and form four spermatids. So meiosis 2 के बाद में four spermatids. And from each spermatid and spermiogenesis, one spermatozoa is formed. That is, from one spermatogonium, four spermatozoa are formed by meiosis. And these four spermatozoa are haploid. A spermatogonium is diploid. So इसमें से चार sperms आते हैं. And when it comes to formation of female gamete, it is called oogenesis. ओवरी के अंदर होता है. oogenesis थोड़ा डिफरेंट रहता spermatogenesis, लेकिन वो meiosis 1, meiosis 2 प्रोसेस रहता. So इधर क्या होता है कि during cytokinesis, that is followed by meiosis, after meiosis 1, the cytokinesis is unequal. So unequal means they are not divided equally. Due to this, one large one is formed, that is cytoplasm, with the most cytoplasm, which is called ovum, and the smaller one is called polar body. It will not survive. Polar body survive नहीं होते. So ovum is rich in nutrients. So this is unequal cytokinesis in oogenesis. लेकिन पूरा process, remaining process same रहता है spermatogenesis. इधर आप देख सकते हैं picture. So this is oogonium, which will form primary oocyte. And this primary oocyte is arrested at prophase 1. It will form after childhood. It will undergo meiosis 1 and form one polar body and secondary oocyte. Secondary oocyte is arrested in metaphase 2. It will form again, undergo cell division, meiosis 2, when it comes in contact with sperm. इसमें भी second polar body रहता है. It will form, which is small, which will result in only ovum. So from one oogonium, only one ovum is formed. From only one oogonium, one ovum is formed after meiosis. So you can see on the right side, there is a simultaneous development of oogonium in follicles. Primary follicle, Graphian follicle. Graphian follicle के बाद में ovulation where secondary oocyte is released. So this is about the formation of gametes. Always gametes are haploid. And due to this haploid gametes, again the chromosome number has come to normal. After fertilization, chromosomes number will come to normal. So meiosis 1, in this meiosis, the chromosome number becomes half. In fertilization, chromosome number comes to normal state. So meiosis helps in maintaining constant number of chromosomes in a species, and it plays an important role in sexual reproduction. At the same time, in genetic variations. So meiosis may be neat me, you can get numerical questions on it is Sambhavana. The first basic point you should remember, in meiosis, from one cell, 4 cells are formed. That is half of the chromosomes. So one meiosis 1, 2 cells, after meiosis 2, finally you get 4 cells. So here gametes are formed, which are formed. Generally meiosis takes place during gametogenesis. This means said, gametes are always haploid. Okay. In all diploid organisms, during sexual reproduction, meiosis will take place during gametogenesis. The male and female gametes are haploid. The reason for haploid is due to meiosis. Okay. So this is the first point you should remember to solve numerical questions. One is from one parent cell, 4 cells. Gametes, the cells which are formed, which have half of the chromosomes. That is, the parent cell is having 46 chromosomes. The daughter cells will have only 23 chromosomes. Half of the chromosomes. Okay. So these two you should remember to solve the questions based on meiosis. Or remember the phases of meiosis in the same sequence. Meiosis 1 may prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 1 may leptotin, zygotin, packetin, diplotin, diakinesis. Remember, in the same sequence, you will get sequential questions. Meiosis 2 may prophase 2, metaphase 2, anaphase 2, telophase 2. And remember, the important steps process occurring in each stage. Zygotin, synapses, packetin, crossing over. Okay. And next, anaphase 1, disjunction. Anaphase 2, splitting of syndrome here. After meiosis 1, 2 cells are formed. After meiosis 2, 4 cells are formed. Okay. And always gametes or haploid which are formed by meiosis. Okay. So there is a meiosis and sexual reproduction are very closely related. Along with these variations, what is the process for variation, that is crossing over? There are 3 mechanisms. Independent assortment, crossing over, and next is random fertilization. So these are the important process which occurs in meiosis. Thank you all. Follow us on social media, SPSVERSITY. We'll meet in the next session.